Thanks very much, Des, for that overkind introduction. It really is an enormous honor to be receiving the Ulysses Medal. Looking down the list of previous recipients, it's rather daunting to be among their number, not least in 2012, the medal was given to Seamus Heaney, a man whose loss is still resonating throughout the civilized world for his humanity, his decency, and his love of truth and beauty. And I have long had a love affair really with UCD, starting when I came in 1975. And even though it was more than 3 hours bus journey back and forth from the north side when I was in the first year, it was a wonderful, eye-opening and liberating time of fun, socialization, and intellectual stimulation. And I met many long-lasting friends at my time in UCD, but I also was taught by some wonderful and inspiring mentors. And although it's invidious to pick out a number of individuals, I just thought I'd mention two names in particular. Maura Beery, who taught biochemistry when I was a young preclinical medical student, and who really brought the beauty and elegance of biochemistry really to light for me and I think several others. And Ivor Drury, now long gone physician in the Mater, who exemplified the virtues of public service and scholarship, and who really saw that the only advances that could be made in patient care would be through the application of high quality and rigorous science. So having had that wonderful opportunity, yes, indeed, I went away, took the King's Shilling and never came back, but I've retained a very strong affection for my alma mater. So this is where I now work, the Institute of Metabolic Science in Cambridge. And there we sort of ask two questions. The first question is why are some people fat and some people thin? And the second question is, when you're fat, why do you get sick? Why do you get diabetes and cancer and other disorders? And we try and understand those processes and then use that information to improve on prevention and on intervention and treatments. And it's important that we do so because diabetes is likely to affect, by about 2030, 500 million people in the world. And it will predominantly do so in countries which do not have the medical infrastructure to deal with the adverse effects of this disease on the eye, on the kidney, and on the nerves to the legs, leading to amputation, blindness, and renal failure in health economies that will be unable to cope with the long-term management of these conditions. And this increase in the prevalence of diabetes has been accompanied by, and possibly, or probably at least largely driven by, a recent change in the prevalence of obesity, which has been unprecedented in the 85 million years of our primate evolution. And our change in the prevalence of individuals who are obese in our species has been dramatic over 100 years. Back in very good survey of American soldiers back in American ex-soldiers, but white northern US males, only 3.5% of them were obese in 1890. And now, practically 35% of that similar age group of Caucasian males in the USA are obese, leading to dramatic prevalences across the various US states. And of course, this has not been driven necessarily by genetic or biological factors, because migration studies, for example, show that one tends to acquire, at least move towards the prevalence of obesity in the place that one has migrated to. So clearly environmental factors are enormously important, and they of course involve the two sides of a very simple thermodynamic equation that determines obesity, that is, the energy you take in and the energy you expend. And over the last 50 years in particular, we've seen unprecedented changes in our ability to acquire excess calories without the need to expend energy in acquiring them. And of course, this is driving the secular change in the prevalence of obesity that's occurred in the last 50 years or so, which may actually be tailing off now as the susceptible people have generally become obese. However, even in Louisiana, which has an obesity prevalence of 40%, at least 25 to 30% of people in Louisiana are lean. Now, what's the difference between people in Louisiana who are lean and those who are obese? Broadly, there's only two possible conclusions. The lean people are either morally superior or biologically different. Now, as someone always prone to be a little bit on the heavy side myself, my a priori assumption is that there must be some biological differences. And actually, the data suggests that that's right, because there is very powerful data, not only from family studies, not only from adoption studies, but from twin studies, identical versus non-identical, of the very strong heritability of body mass index, of indices of adiposity. And be very careful what you mean by heritability. Heritability really means if I took you all out of this room, put you out on the hall outside, lined you up from the leanest to the fattest, and then picked one of you out from the low end and one from the top end and said, what is causing the difference between you and you in your amount of fat you've stored? And all the data suggests about 70% of that difference is down to the genes that you've inherited, and the rest is down to other environmental factors. So it's a larger percentage than people are intuitively inclined to think. And that hasn't gone away. Those studies, there's at least 20 twin studies, all doing that. And you can see just a graphical illustration here of sisters, 4 pairs of sisters, all born on the same day, but just happening to share 50% of their genes because they're dizygotic twins. And they come in all shapes and sizes like brothers and sisters do. And then these are 4 pairs of sisters born on the same day, but sharing 100% of their genes. It's hard to look at that and say, I don't think genes are terribly important in determining your body. And it's a pretty visual, impactful example of how powerful the genes that we inherit are in our physiognomy generally. And that heritability hasn't gone away, even though we're in the midst of an obesity epidemic. Jane Wardle in London did the similar study in twins in London just a few years ago, in 11-year-old twins, and she calculated the heritability around 70-75%. Again, in a very large twin study. And we even have remarkable studies done by Mickey Stunkard. In Sweden, in the 1950s, the sexual revolution preceded the contraceptive revolution, and there were lots of babies born out of wedlock. And Sweden being a nice middle-class place, lots of parents wanted to adopt children, but they didn't want to adopt 2. And the Swedish social system decided it would be a wise thing to separate children at birth, give them to different parents, and not tell them that they were identical twins. Now, this was not an experiment, this was a social decision. But it led to the situation which allowed a Philadelphia-based psychiatrist, Mickey Stunkard, to realize that there were 30 to 40-year-old people in Sweden who are now being revealed to them that they were identical twins. These people knew that now, but he was able to go to the data sets and ask, what was the concordance between your adult body weight, your tendency to be fat, and the family that brought you up, the one that you fed with and you played with and nurtured you from birth or for very early after birth, and the identical twin that you'd never seen, didn't even know existed. And the correlation with the family that brought you up was precisely 0. There was absolutely no influence, or at least no correlation. It wasn't just low, it was 0. Whereas the correlation of your BMI with your identical twin that you'd never seen was 0.7. So a very powerful influence of these genetic factors on body mass index. So that may seem a bit depressing. One of the issues about genetics is people think genetics is immutable. I always envisage it as like understanding the wiring diagram under the car. It's a good place to start, so then we can start doing something about it because we can understand how the car works. And so I'd just like to address the question of why, if I was interested in these diseases, why would I bother doing human genetic research? There's 2 reasons for doing genetic research. Firstly, it might be useful. Now, how might it be useful? It might be useful because we could give people, if we knew what was going on really at a molecular level, we could give people meaningful diagnoses where previously they only had vague nostrums, vague terms about their condition. We might be able to tell parents of children or patients themselves a bit more about what was likely to happen to them if we understood precisely the mechanism of their disease. We might be able to take apparently healthy people and give them an assessment of how high at risk they were of particular diseases and how to alter their behaviors, perhaps to avoid ill health in the future a bit better. And we might be able to take our therapies for metabolic disease and target them to the disease mechanism so that we can actually have a better and more precise and effective result. But of course, the dirty little secret of science is that many of us do it, and many, as I'm sure in this audience, do science not only because it's useful, but because it's interesting. And human genetics, of course, can be very interesting. It can actually teach us about how we evolved from lower organisms to the organisms we are today. It can teach us about how we developed from a zygote into a large adult organism. And importantly, it can teach us about how the adult organism functions. That is, the old term, physiology. And physiology is really important, and really important for medicine, because what we do when we use most effective drugs, antihypertensives, we tinker with the control nodal points in physiology. And human genetics can be very useful in teaching us what those control nodal points can actually be. And actually, if we understand human physiology better, that can make the interesting into the useful in terms of a pharmacotherapy and drug development. So when you decide that you're using genetics to probe physiology, then you're talking to an audience largely of basic scientists. I often feel that I need to explain why it's a terrible idea to study human beings. Human beings, we cannot, thankfully, control their genotype or their breeding. They have unconscionably long generation times. We cannot produce breeding colonies of massive to find out phenotype. And we cannot manipulate the genome in a tissue-specific or a temporally controlled manner. So we can do all of these in model organisms and get beautiful insights into the physiology of control of any system. However, there are reasons to embrace genetic studies in Homo sapiens. It is, after all, the species in whose health and well-being we are most invested. The physiology of man is not always paralleled by the physiology of lower organisms, even ones as close as mice. We, for example, recently shown that neurokinin B, patients who lack neurokinin B, human beings who lack neurokinin B, never go into puberty. It's a complete gate for pubertal development. And it is completely redundant in the mouse. It has no impact on the reproductive function in mice. Importantly, rare variation in human beings is more often brought to our attention than rare variation in other species, because we as a species take our rare variation to trained observers, i.e. doctors. And doctors communicate with each other through journals and through the internet. And we have, as medical researchers, access to this vast range of genetic variation in the human species that can be very illuminating. What again is often not emphasized is that because we've had to apply physiological tools to humans in clinical settings that involve real therapeutic decisions, we've made sure that our tools for human investigation are very well validated and very well replicated and under continuous quality improvement. And we actually have very good tools for measuring things in humans that often have not been as well validated for the small organisms that we sometimes play with. And then finally, of course, the wonderful thing about humans is we can explore things that are much more difficult to explore in lower organisms, i.e. we can talk to our patients and we can talk to people and we can get insights into emotional and cognitive and other aspects of biology that are really inaccessible or less accessible in lower species. So, having tried to convince you that genetics is a reasonable thing to do and human genetics is a subtype of genetics that has its own particular power, I'm now going to tell you that there's basically two broad ways that you might use human genetics. And I'll parody it a little bit as saying, there's the butterfly collector, the individual who studies extreme, rare, but very illuminating and informative rare variants of major effect in the population. Or there's the multinational owner of multinational industrial sweatshops who study hundreds of thousands of people with very small common genetic variation of very small impact, which affects large swathes of individuals throughout the world. And both have their value. And I'm going to talk mainly about the butterfly collection because that's one that we've had a particular useful results from. But that is not to say that the other approach to genetics has not been helpful and illuminating. So we started to study obesity about 1992-3, really based on a single patient I'd seen in a regular clinic I was doing at Addenbrooke's Hospital. She was presented to the endocrine clinic with reactive hypoglycemia, but had lifelong severe obesity. And to cut a long story short, we did some detective work over 2 or 3 years in an era when it was not easy to do the molecular biology, and we had to clone the human gene before we found the mutations in it. But we did find an underlying cause of that woman's syndrome, which included severe lifelong obesity. And that suddenly made me aware that obesity was not perhaps just a simple social problem. I'd been thinking about it as lazily as everybody else I think had been thinking about it previously. And around that time, there had been a number of wonderful discoveries in naturally obese mice where the molecular basis of their obesity had been uncovered. So that sprung us into action in this area, and with some wonderful postdocs and fellows at the time, and Sadaf Faruqi, who's now become a leader of our studies in the genetics of childhood obesity, we started looking for obese genes. Now what I mean there is we decided to look where the children were extremely obese. We didn't look for common obesity. We wanted the obesity to be very early onset, occurring within the first couple of years of birth, and very severe, where it seemed to be very unlikely that any parent would deliberately, or even not deliberately, be overfeeding their children to result in that sort of obesity. And by studying that subgroup of individuals with obesity, we rapidly found a number of genes. And those genes are genes that really are causative. They're not just associated. When these genes are broken, if you know, when we have mutations that disrupt function, you are almost inevitably obese. They have very high penetrance when the genes are present, either in homozygous or in some cases heterozygous mutations. And without going through all of them in detail, I'll say I think we discovered two things. And the first thing we discovered is that all of these genes were highly expressed in the brain. They weren't expressed in fat or muscle, they were brain genes. They were genes that were having their primary expression level in the brain, and particularly in the hypothalamus, the area of the brain known to control a number of homeostatic processes in the organism. And the second thing we did is we actually didn't just leave it at that. We took these children into our clinical research facility, and we in detail evaluated their food intake, their energy expenditure, and the contributory physiology to their obesity. And in every case, all of these children, the main factor driving their obesity was that they had an uncontrolled appetite. They had an increased appetite and a decreased satiety. They were unable to sense when they'd eaten enough food, and they got very hungry shortly after consuming a lot of food. And they were very different from their siblings who did not contain the same gene but who had grown up in the same family. So this was really, seems very simple, but actually it's quite challenging, and still people find it hard to think that something can be both genetic and behavioral. So this was a gene. We could see it, we could measure it. Indeed, in the case of melanocortin-4 receptor mutations, which is 50,000 people in the UK with this disorder, it's commoner than cystic fibrosis. We can take those mutations, put them in the test tube, measure how much cyclic AMP is being generated, and predict how much someone will eat when they sit down in front of a meal. So this is a single gene having a powerful impact on what we think is a voluntary process, but it really has a very powerful lower brain driver to it. And of course, we had the great good fortune and wonderful excitement of being able, in a very small subset, sadly, of cases, particularly only so far those with congenital total deficiency in the adipocyte hormone leptin, to be able to restore this. And now in 25 or so children, all of whom have completely reversed their lifelong, life-threatening obesity, which has actually in some cases has already killed their older siblings before we could. Many of these children don't make it into mid-teens because of respiratory complications of the severe complications of their obesity. So this was life-saving therapy, albeit for a very small number of children. I think a small experiment we did led by Sadaf with Paul Fletcher and Ed Bullmore as our neuroscience colleagues, I think it was quite an illuminating experiment, just bringing together this whole sort of the ridiculousness of the mind-body dichotomy, really. What we did here was we took two adolescents who presented, 14 and 15, with congenital leptin. Well, they'd presented all their lives, but they were diagnosed in their adolescence. And they agreed to have functional magnetic resonance imaging studies of their brains so we could look at the effect of leptin therapy on how their brains were changing when we gave them therapy. And I'm not going to go through this slide in complete detail, but what I'll say is we showed them pictures of food, essentially. Food and non-food, and sometimes appetizing food and sometimes non-appetizing food. Essentially, when you show these leptin-deficient children pictures of food, the areas of their hypothalamus concerned, the areas of the brain concerned with the reward, the nucleus accumbens and the putamen, and other areas that really light up when you show a cocaine addict the paraphernalia of cocaine administration, lit up like a beacon in response to the visual stimuli of food, dramatically increased above normal. And when we scanned them later, 3 days after the administration of leptin, before they'd lost any weight, this little molecule, which is just produced from fat cells, only produced in fat cells in the body and going to the brain, had totally reversed this abnormal visual response to food. So here we have an example of how our endocrine milieu is having a profound effect on how our brain is perceiving visual stimuli related to food, and showing that our sort of higher hedonic centers controlling appetite are very intimately associated with these endocrine and lower center and the information coming up from the body. So have we got any more? I mean, have we come to the end of our studies of genetics of obesity? Well, there are plenty of very obese children in our 5,000 patient cohort. Now, these are from all around the world. These children are sent us, we're asked to consider them from places all around the world. We take the samples. We have a gene-first approach, a gene and biochemistry, and then if we find something, then we invite parents and children to come to our facility. We can't see all of these children, but there are still many who are just as obese as the children that we found the genes in, that we don't know the cause yet. And we continue to find in the last couple of years, for example, we just found that mutations in a transcription factor involved in hypothalamic development of a paraventricular nucleus in the event is a relatively common, around 1% perhaps of our cohort have got loss of function mutations in SIM1. A much smaller number have got mutations in an accessory protein for the melanocortin 4 receptor, and through a variety of approaches we found that deletions of a molecule SH2B1, which is downstream of leptin and insulin signaling in the brain, also causes an obesity syndrome. And that's not the end of it, because what we've been doing in the last couple of years is in association with Inés Barroso at the Sanger Institute, which is the major Wellcome Trust Genomics Institute. We're currently plowing through data from 1,000 we've sequenced every exome, all of the encoding DNA in 1,000 severely obese children, and comparing those to the control data set. And we're already finding unique variants which are only found in the obese group, which are very powerfully statistically associated with obesity. Many or several interesting G protein coupled receptors that we're working on at the moment, and may form therapeutic targets, we hope, that were previously unknown. And I'll just show you one example of work that's in press related to another obesity gene that we've recently worked on. So back in 1995, the Horvitz lab and the Rubin lab in worms and flies, respectively, discovered a new element of the classic RAS-MEK-RAF-MAP kinase cascade, and they discovered a gene which they called kinase suppressor of RAS, KSR. Effectively a scaffold protein which has complex effects on signaling through this. It's both necessary for normal signaling, but it's also involved in the feedback attenuation of signaling. So its disruption, the effect of its disruption ultimately on that signaling cascade is somewhat unpredictable and very tissue specific. So there are two genes encoding two isoforms of two individual separate genes for KSR, KRS1 and KRS2. And KRS2 was first identified by the Lexicon lab through a high throughput genomic screen, where they found an obese mouse the responsible gene was KSR2. They didn't do much phenotyping on that. It was, to say, a high throughput approach. Then Rob Lewis in Nebraska studied the mouse in more detail. You can see this is the knockout mouse, this is the wild type mouse, and confirmed that it was obese, but showed that it had a defect in energy expenditure predominantly. It was really slow at burning off fat. It also had a defect of AMP kinase signaling and was very insulin resistant. And then the Powell lab did a bit more work and showed the mouse was also hyperphagia. So perhaps two different defects contributing to the obesity in the mouse. So it's a very good candidate gene, an obvious one for us to study, and so we studied it. And we studied it in 2,000 of our goose cohort and almost to the 1,500 of controls, and found a highly significant p less than 0.0025 enrichment of point and nonsense mutations in KSR2. Some of these mutations are frame shifts and premature stops, and as you're seeing here, there is where are we now? Yeah, phospho-MAP kinase. This second line here. It just slightly fails to line up here, but this is the phospho-MEK blot, and there's absolutely no increase in phospho-MEK in response in cells that lack that have the mutant receptor, mutant KSR2 present. And similarly, many of the point mutations have attenuated signaling to downstream MEC or ERK signaling. Now, many of those point mutations are at the surfaces of the interfaces with MEC1 or BRAF and would be predicted to disrupt that complex. Again, what we do, as well as doing the cellular biochemistry, we like to try and combine that with detailed physiology in the humans. So we brought the children and adults with these mutations back into the lab. And this, for the first time, really, we're able to demonstrate a human obesity gene that had a substantial and detectable effect on reducing your energy expenditure, your basal metabolic rate. So this is a gene which, as well as causing a degree of hyperphagia in children, but not in adults, does have an effect on lowering energy expenditure, and therefore, and very simply, as well as excess energy in, this gene does influence energy out, and those two together contribute to the insulin resistance. This is the insulin levels after a glucose tolerance test in these patients compared to equally obese controls without mutations in this gene. You can see how spectacularly hyperinsulinemic this group are. Now, Sadaf made the interesting observation in talking to these patients that many of them had been given metformin as children because they presented with a skin lesion associated with insulin resistance called acanthosis nigricans, and they seemed to have an unusually robust weight loss response to metformin. And we found that intriguing, particularly given the Lewis observation that AMP kinase activity was deficient in cells in animals lacking KSR2. So we looked at cellular energetics and using the seahorse system, examined oxygen consumption rates in C2, C12 myocytes expressing wild-type KSR2 and the mutants. And you can see that there was a marked effect of these mutants on glucose oxidation, and also, except for one mutant, on palmitate oxidation. But intriguingly, if we tried to rescue this by metformin, we could actually show a significant, really a restoration of response to normal in these mutant cells with metformin. So we hope we may have identified a subgroup of obese children and adults who may have a particularly robust response to metformin. We know that metformin only causes, on average, a very small weight loss when it's used, and as it's used in many millions of people worldwide. But this may be a subgroup of individuals where the effect is particularly robust. Don't quite understand exactly why this works, but the link is intriguing and potentially therapeutically useful. I'm just going to show you one slide related to the common obesity, the sort of industrial sweatshop obesity. What essentially I see that type of genetics is doing is looking down from space onto the landscape of a disease and just picking out the broad outlines of what is the underpinning biology. So if you look, for example, at diseases that you think might be inflammatory in origin, you see lots of signals for in genes involved in inflammation. If you look for type 2 diabetes genes, you find lots of genes that are associated with beta cell dysfunction. When you look at obesity and all obesity-related parameters, what you find are genes expressed in the brain. And many of them are the same genes that we find are mutated in the severe syndrome. So there's a considerable overlap between the genetic architecture of the common variation present in this room with these rarities that we find when we study very severe obese children. But when we ask the question, do the same genes determine how much fat you have, or where you put your fat? And so this is a set of genetic variants that's associated with waist-hip ratio, with whether you're centrally obese or whether you're peripherally obese. And there's almost zero overlap. And most of these genes are expressed in fat, in muscle, and effectively under the neck. So in very simple terms, how much fat you have and store is an above the neck problem, and where you put it is a below the neck problem. So in conclusion, we can go back to the whole useful and interesting questions at the beginning and ask about severe childhood obesity. Have it been useful? Well, we certainly can give people a more accurate diagnosis and prognosis with many of these diseases. And we can treat dramatically one rare form of obesity. Sadly, the problem about leptin is it's very effective when you go from no leptin to some leptin, but it's rather ineffective going from some to more. So the shape of the dose-response curve militates against its general use. In common adult obesity, sadly, we haven't really got a clinically useful risk predictor, nor can we use information genetically in any useful, immediately clinically applicable way. Has it been interesting? Well, that's really a question of scientific taste, and you can all answer the question for yourselves because it's in the eye of the beholder. But I think it is interesting that the work has led to the thought that, mostly doctors and scientists 20 years ago didn't even think about obesity at all. If they did, they thought of it as possibly a disorder of metabolism and of energy balance. And I think with this body of work and others going around, we are beginning to think of it more as a genetically predisposed, neurobehaviorally based trait, which makes people differentially susceptible to the obesogenic environment that is being produced by our current societal pressures. And the broader question of obesity requires a different lecture. The audience today, I know, is predominantly basic science, and so I'm giving you more of the hardcore blots. But there's some fascinating questions about how we best address obesity in a more societal and context. But that's a question for another. I'm happy to answer questions if that's appropriate after the talk. So I'm going to move in the second half of my talk from above the neck to below the neck, if you like, and ask the question of how do we get from obesity to the diseases that obesity is associated with. And it's important to realize that it's not intuitively obvious why carrying around 20 excess kilograms of triacylglycerol in fat cells hanging out under your skin, why that leads to diabetes, insulin-resistant diabetes. It's not intuitively obvious, because actually fat's pretty irrelevant to glucose control by and large. If I give somebody here a 75-gram oral glucose tolerance test, only a gram and a half of that glucose will end up in their fat tissue. Where it ends up is in liver and muscle, and that's where insulin is working to control glucose homeostasis. So we have to understand, how does obesity lead to insulin resistance in liver and in muscle? While obesity is essentially a problem of too much fat. So how do we explain that? Well, there's a kind of broad consensus emerging in the community, which I can never see a consensus but want to prick it. And the consensus is that the culprit in this is the big bad fat cell. So that the fat cell gets bigger and larger, it gets very enlarged, it starts producing stuff. It starts producing a whole range of adipocytokines, things that are both immunologically active and have other adverse effects on surrounding tissues. And it's these guys, these adipocytokines, etc., and the inflammatory state of obese adipose tissue that somehow, either through a paracrine or endocrine mechanism, impair insulin action in liver and muscle. However, there's a more long-standing and older view of the problem of how you link what effectively obesity is. Another way you can define obesity, I mean, it's almost a self-fulfilling definition. Obesity only occurs when you have a sustained positive energy balance. In other words, when your energy in is chronically greater than your energy out. So that is, by definition, what obesity is. So if you have a sustained positive energy balance, actually, the safest place to keep that energy surplus is your fat cell. In fact, your fat cell is your friend, not your enemy. It is the place that you really want to store your excess. That's what it was designed to do. And one view is that we all have a limited capacity to store, or some sort of limit to our capacity to store fat safely in fat cells. And we all reach our limits at different levels of fat storage. And that what actually is happening is that you're reaching the link between obesity and its adverse consequences are when your nutrients excess is being redirected to places where it shouldn't be stored, i.e. liver, muscle, pancreatic beta cell, vascular. And it's that is actually causing the metabolic mischief, and not these inflammatory issues. Although they're not mutually exclusive hypotheses. And I'll just show a brief one-slide summary, which I think, to me, provides quite compelling evidence that we have to think that second mechanism must be relevant. Because here we have two genetic diseases that we described over the last few years, led by my colleague David Savage in the lab in Cambridge. David's now a Wellcome Senior Fellow and reader and runs his own group. We work closely together. And these are two genetic conditions that were not described until David and I and the other group found them. And these are genetic conditions which only affect the surface of the single triglyceride droplet in the fat cell. And they're regulatory proteins that are concerned with the regulation of lipolysis at the surface of the fat cell. And if you are born with a defective member of one of these families, you develop the full panoply of syndromes of type 2 diabetes, insulin resistance, fatty liver, solely because you cannot safely control and keep and regulate the creation and release of fat. No need to imply any inflammatory involvement. This is solely a metabolic flux problem from the adipocyte, leading to the full range of syndromes. And yet I'm struck by the fact that I see a group of patients with another type of fat disorder, which is called acquired partial lipodystrophy, which is an autoimmune disorder, which is a hugely inflammatory condition, which sometimes destroys half your body fat, taking fat off the face, on the limb. These people have aggressive inflammation in fat, so much so that they actually destroy their fat. And they have no metabolic complications, so long as they retain enough healthy fat in the lower half of their body. And I'd just like to put that out there in terms of how we think about this whole inflammatory hypothesis of obesity associated in insulin resistance, which so dominates conversations at the moment in the field. The last, I'm going to show you a few pieces of work now where we move from the fat cell to the target tissues in liver and muscle, because that's where insulin is really having its final actions. And we know a lot now about how insulin works. It works through PI3 kinase, activating activating the AKT2, which is a key metabolic regulator both of glucose transporter translocation in muscle and fat, and in suppression of gluconeogenesis in liver through FOXO1 and other mechanisms. And we know a lot of that from work in cells and animals, but human genetics has played a surprisingly small role in elucidating these pathways. And there's been a bit of a gap in trying to understand whether which elements of these are specifically important for humans. And we've had a few, I'll just show you 3 examples, recent examples of where we think we have managed to fill in that hole. This is just a single rare patient, but an illuminating one. This is a young girl whose mother and father are both Nigerian. They're both over 6 foot, and she's small and lean. She's not lipodystrophic, but she presented with polycystic ovary syndrome, which often happens when you have severe insulin resistance. And her testosterone level, for any clinician in the audience will know, that's really a male testosterone. It's 10 times higher than normal testosterone from the abnormal ovaries, with massive levels of fasting insulin. So hugely insulin resistant young girl. And her parents were pretty much entirely normal. So in this modern era, what we can now do is whereas most of my life was spent trying to do clever detective work, trying to think about what genes might be involved in someone's syndrome, we now, for a few £100 and a helpful bioinformatician, can do dumb genetics. We can actually do whole exome sequencing, and we can look, and particularly when we have the parental DNA, we can look at any genetic abnormality that's present in the offspring and not in the parents is likely to be a candidate for the causative lesion. And in this case, exactly that. We found really only one compelling new defect. Remember, we all have de novo mutations, about 7 in our exome, about 7 you've got about 7 mutations in your DNA, 7 variants that affect the sequence of your proteins that were not present in your mother or your father's genomic DNA. Of course we have, because we wouldn't have evolved from slime molds if we didn't. This is the energy, this is the engine room of evolution. This is without that genetic variation, some of it is harmful, some of it's neutral, and some of it might be beneficial. So that's how Darwinian evolution works. But this girl was unfortunate for her particular accident to occur as a stop codon in the P85 alpha subunit of PI3 kinase in a heterozygous fashion. And this mutant form was actually expressed in her cells, both her EBV lymphocytes and her dermal fibroblasts, as a pretty abundantly expressed. It was clearly not being cleared as fast, so she had abundant amounts of this P85 in her truncated form in her cells. And in her dermal fibroblasts, it looked like it was affecting certainly the ability of P85 to interact with phosphotyrosine residues, both in her cells and in transfected cells. In that phosphotyrosine, clearly with insulin stimulation, P85 alpha is designed to bind to phosphotyrosine residues and transmit signals, and hers clearly was unable to do so. It took some pretty subtle cellular biochemistry led by a wonderful postdoc in the lab called Nuno Roca, who made a lentivirus vector which we could control with doxycycline, And you can see that the effects on downstream signaling are actually rather subtle. But I'll just draw your attention to this area here. Here we're inducing, this is no doxycycline, this is doxycycline. It's inducing the expression of the mutant P85 and impairing at low levels, significantly impairing signaling to phospho-AKT. So this mutant P85 is really having a dominant effect and interfering with signaling even when present at low levels within the cell, and having subtle effects on impairing the adipocyte differentiation when put into 3T3L1 adipocytes compared to wild-type P85. But we didn't have a single other patient with this mutation, or indeed mutations in this gene, so it's very hard to prove causality. And this is where it's very helpful to do bidirectional translational research. So to really prove this was causal, we made a mouse. So we took the mutant and we put it into the murine genome, and have just recently successfully, Jan, this is very early, just as really very early data. Already we're seeing that the younger mice that we're generating, the wild-type mice, the mutant mice are significantly smaller, like the patient was. And when we start doing oral glucose tolerance and insulin tolerance tests, the mice are significantly, in fact, severely insulin resistant. There's no response to the administration of insulin. They're glucose intolerant. So we think we've probably generated a very useful mouse model of this girl's disease, and this will allow us to use therapeutic and other trials to look at ways of abrogating in an animal model before we bring it back into the patient. The second example is a fascinating sort of new type of human metabolic disorder. This is a young boy I was called down to see, or asked to see, about 10 years ago in London, because he was admitted to hospital with series of fits associated with hypoglycemia. He could not, at the age of about 1, he could not be starved, or he could not be left overnight because he'd be found fitting. And he was fitting because his blood glucose was less than 1. But his insulin was undetectable at the time that he was fitting. So it was not being driven by discernible and detectable insulin. And the other features you might notice in these, you can see that he's needed a parenteral feeding, and so have these two. So these children require overnight feeding directly into the jejunum to prevent these hypoglycemic attacks occurring overnight. For those of you who are clinicians, you'll also know it's a very fascinating situation. These children are not symmetrical. They have left-sided hemihypertrophy. Their left sides of their body are slightly larger than their right, and they're not mosaics for the mutation. At least two of them are not mosaics. So it's not a mosaicism phenomenon. It's a very interesting and is yet unexplained phenomenon. And what they all have is we did whole exome sequencing, and again, parents completely normal at this gene, and what we found is a mutation that was occurring presumably in either the mother or the father's germline, and present in there. And it was the same mutation in all three unrelated patients in what was called the pH domain of AKT2, which is that key metabolic signaling kinase downstream of PI3 kinase. And we actually do understand this now at atomic detail, because we know exactly what the pH domain does. It binds PIP3. When PIP2 is phosphorylated to PIP3, the pH domain of AKT2 docks beautifully with PIP3. And this mutation changes the binding face of the pH of the pH domain such that it's agnostic as to whether PIP2 or PIP3 is there, so that it actually just goes to the membrane anyway and is activated in the absence of insulin. As you can see in this diagram here, in transfected cells, not sure if it shows up on that light, but here with normal cells, if you can see in the red, I can't really see much here, but in normal cells, it's intracellular. When stimulated by insulin, it goes to the plasma membrane. But here in the mutant, even in the absence of insulin, this kinase is at its active place. It's at the plasma membrane. And being at the plasma membrane, it's active. And we know it's active because we've got a very nice marker of cellular insulin action. That's the transcription factor of FOXO1, which even with a whiff of insulin moves rapidly out of the nucleus. And we can see that in wild-type AKT2, FOXO2 is in the nucleus, and then rapidly in green here, when given insulin, it goes out of the nucleus. But in the mutant form, even in the absence of insulin, FOXO is out of the nucleus. So insulin is acting even though there's no insulin there, because AKT2 is mimicking that. So we have a new type of disorder. And it does obviously suggest a therapeutic opportunity, as AKT inhibitors are being developed for the treatment of cancer. So we may be able to get to a stage of using this therapeutically in these children. And the fascinating implication of AK2 in determination of left-right asymmetry, which is rather unexpected. And then finally, you may find this rather a shocking picture, but Mandy, who's the name of the patient, is a very great advocate for this condition, has insisted that we help her make TV programs about it, and wants to be known about it as possible. And Mandy was born with 2 very big legs, and her legs never stopped growing all her life and continued into her 30s. And I was asked to see her really because nobody really could understand what was going on, and she looked rather lipodystrophic in the upper half of her body. And we thought, this is very odd. This is the size, this is her right foot, an example with the hand of a nurse. So you can see how absolutely enormous these legs become. She, because of the compromise to the size of her left leg, eventually she had so many ulcerations and infections, she had a below-knee amputation on the left side. So in order to try and work out what might be going on here, we did again what was a very simple experiment. We thought we would just take cells from the top half of her body and cells from the lower half of her body and do whole exome sequencing and see what differences there were between them. And that's what we did. And we found that actually a very obvious candidate. We found that the PI3 kinase, again, that kinase that's upstream of the AKT, she had a classic mutation in that had previously been described as an oncogene in various cancers. And we found, and it was essentially in fact, it was discussed today. It was on one of the posters in the Conway, used as a classic oncogene, and in the kinase domain of P110 alpha. And we collaborated with Les Biesecker, who a NIH investigator who studied the Proteus syndrome, the one that the elephant man had, and Biesecker had recently described AKT1 mutations as causing the elephant man syndrome. And we found that his patients who didn't have the elephant man type syndrome, but another regional overgrowth syndrome, they all had mutations of a very similar nature in P110 alpha. And you can see that this isn't just a theoretical increase in activity. With Mike Wakelam from Babram, we directly measured PIP3 levels in the arm and the leg, for example, here, normal levels in the arm from the patient from fibroblasts, very elevated levels. So this was biochemically active, pushing the PIP3 levels markedly up. So again, what we like to try and do is think therapeutically about how we find these things, whether we can do anything about it. And there are numerous points that you could interfere with this action in patients. But there's really only one available inhibitor in this signaling cascade that's actually in the clinic and has been in long-term use for some time, and that's sirolimus or rapamycin, been used for immunosuppression in renal transplantation for a long time, but is quite a powerful inhibitor of the mTOR signal, which at least is responsible for some of the downstream growth effects. Certainly the cellular size effects, S6 kinase, its downstream mediator, is a powerful regulator of cell size. So because Mandy really was keen on having some therapeutic intervention, and even though this drug has potential side effects, because her mobility would have been, to lose the other leg would have really fatally affected her mobility. She was very keen to have a therapeutic trial of this. So we did take her cells and see if mTOR inhibition would work, and in her cells it works beautifully. It completely reverses the growth, this downstate, certainly the S6 kinase activity in her cells, and indeed the AKT2 is pretty much reversed by the administration of this drug, which is the same sort of just the blots are just shown here. You can see her mutants with the drug, complete suppression of P70 S6 kinase phosphorylation. So this drug works in vitro, certainly, and it works in her cells. So we wanted to develop some measurement techniques, because measuring limbs like Mandy's has never been very easy, has never really been done before. So with our imaging colleagues, developed volumetric techniques to make sure we knew what we were doing, and started treating her and started measuring that limb. And we started on very low doses, because this is a potential immunosuppression and is an immunosuppressant but this is relatively low doses. And it's an N of 1 trial really on the grounds of clinical need, an N of 1 patient trial. But I think it's encouraging at least that we may have stopped the progression and at least done something to arrest the problem with this in this patient. And there are many more patients out there. The unmet need becomes as soon as something becomes possible, then the number of referrals keep flooding in. And there's a much larger number of these patients around than we actually thought. What's fascinating about these is despite this mutation being a well-known oncogene, some of these patients are in their 50s and not one of them has cancer in the affected area of their body. So alone, this well-known oncogene is not sufficient to cause cancer in humans. These are developmental mutations occurring at, in Mandy's case, probably about day 12 of the embryo, affecting the segment of her body that was developing at that time. Some of the smaller areas are when a ray of a limb is being affected, that's a much later mutation. But in none of these alone, these are not sufficient to cause cancer. So as the so-called idea of driver mutations, well, you need lots of drivers, I guess. One driver isn't enough. So I'd like to conclude by saying that generally we've viewed over the years, and I see some of my old clinical genetics colleagues here, and I think, Andrew, it would be fair to say that when we were training in Dublin, clinical geneticists were out the porta cabins. The sole clinical geneticist was out in a porta cabin at the back of the hospital, and they weren't given. They had books of dysmorphology, and they weren't given prescription pads, and it was considered really not part of mainstream medicine. But what we've seen over the last 20 years is how profoundly genetics has come in to be important for all aspects of what we think of as mainstream medicine, whether you're interested in height or cholesterol or calcium or diabetes. And these disorders are probably continue to be unrecognized in mainstream clinical practice, but when you do recognize them, they might be amenable to specific treatment, and they're certainly likely to be physiologically illuminating. William Harvey, the discoverer of the circulation of blood, wrote to a Dutch physician. He actually wrote it a week before he died, and I find this quote very helpful. He said, "No better way to advance the proper practice of medicine than to give your mind to the usual law of nature by careful investigation of cases of rare forms of disease." We don't notice things' usefulness till we are deprived of them, and by finding individuals with genetic loss of function mutations, we can really appreciate the normal physiology and their role in normal physiology. So I'd like to thank particularly my colleagues and collaborators who I hope I've thanked, particularly obviously my longest standing fellows and now senior colleagues, Sadaf Faruqi, David Savage and Robert Semple, and all our wonderful postdoctoral and other colleagues that have worked with me over many years. The referring physicians around that really are necessary for our work, our funders, but particularly the patients and their families. And with those in mind and the sorts of suffering and problems that they've had and the frustration they've had in having diagnoses and people to understand what's been going on, I'd like to leave you with a quote from Seamus Heaney from a wonderful poem, The Cure at Troy. But that's not really the last thing I'd like to leave you with, because I know that several of my friends have been teasing me about my recent meeting at Windsor Castle, and I thought I'd just leave you with that. So thank you for your attention.